So I would like to request Dr. Mukesh Meghwanshi to kindly come on the stage to co-chair the session with Professor Ratnagiri. I also like to request Dr. Sagrika Biswas to kindly come on the stage to co-chair this session with Professor Ratnagiri. I like to request Professor Ajay Kumar Sharma sir to kindly chair the session with Professor Ratnagiri. So we are now starting the technical session. We have Professor Ratnagiri and Professor Ajay Kumar Sharma as our session chair, and Dr. Mukesh Meghwanshi and Dr. Sagrika Biswas as session co-chair. The first lecture is from Dr. Ritesh Agarwal, Head of Indira Fertility Academy. I would like to request Dr. Ritesh Agarwal and Mr. Naval Shah to please come on the stage for their distinguished lecture. Dr. Ritesh Agarwal, Mr. Naval Shah.
Hi, good afternoon, everybody. First of all, I would like to congratulate MLSU and Dr. Tikam for their wonderful session. This session on biotechnological and environmental and chemical sciences. So this is a great initiative, and I can really see so much of sleepless nights they must have gone through, the whole team, before coming up with such a nice event. Second, my deep heartfelt thanks to their team for us an opportunity to introduce ourselves as a company. So we are basically Indira IVF. I know, although it's entirely a different field from what you must be expecting before coming to this auditorium. But believe me, whenever we talk about technology, so it is not limited to any particular segment. Rather, it is a very broad thing. So now biotechnology, now even infertility treatments, they definitely depend a lot on biotechnology students, biotechnologists for their treatment. So in the coming slides, we'll be going to show you how you can contribute to into infertility industry. Although it's a medical field, you definitely can contribute. Along with me, I have my colleague, Mr. Naval Shah. He's a head of the audit operations with Indira IVF, and I am looking after Indira Fertility Academy, which is particularly working into academics.
Thank you very much, Riteshwar, and thank you, organizers, especially Dr. Tikam. It was a great to have him since day one, and we could see the way he was speaking and delivering to make this event a successful and a big one for all of you. I think his dedication for students and university is definitely appreciable. So thank you, Dr. Tikam, for also along with that giving us opportunity.
Thank you, Dr. Mr. Naval and Dr. Ritesh Agarwal, and I am here for you, and I extend all my support. You are launching very, very technically sound courses for the master students, and I would definitely extend all my support to you in your all future endeavors also. Thank you very much for coming here.
I'll just very quickly brief a bit about infertility. So that is how, first of all, what is infertility? Infertility is inability to conceive after 1 year of married life and with having regular relations with the husband and wife. But if they are unable to conceive, there could be enormous reasons for infertility. But enormous reasons for the infertility, which broadly they are 50% because of the male and 50% it is because of female. So male infertility is basically because of poor sperm quality, be it motility, morphology, or the counts, sperm counts, or even the DNA content of the sperm. But if we talk about the female, the list is endless. So it could be the poor quality oocytes, it could be poor quality of endometrium, uterus, or any ovaries, any blocked fallopian tubes, endometriosis, and the list goes on like this. So what is our role? So as an IVF center, it's a huge banner. So it covers all the infertility treatments, and IVF centers, very popularly known as test tube baby centers. You must have seen so many test tube baby centers. They are in all the cities. So they particularly deal with infertility problems. So in a test tube baby center, basically it depends on two pillars. One is the clinician, who is an MD or MS gynecologist or a DGO gynecologist, and another is a embryologist, a clinical embryologist. Earlier, the clinical embryologists were all life science students who were from various fields like microbiology, biotechnology, zoology, botany, all these fields. But now, with new ART guidelines, I'll just explain you. So this is the work which goes in a IVF treatment. So the doctor first prepares the female for egg retrieval. So they are going to take out multiple oocytes. Then they give those oocytes to an embryologist, which is a life science postgraduate and trained enough to handle all these procedures. So those technicians, those biotechnology embryologists, they inject each sperm into oocyte to fertilize them, and they fertilize. They take them to day 5. 5 days they keep it in the incubator, and when the embryo is formed, they either transfer the embryo into the uterus or of the lady, or they freeze it for further usage. These are various procedures which are done by an embryologist. I won't go into details. And so basically, clinical embryologist is a biologist or a scientist. They are really working parallel to the doctors. They are actually considered, they are highly respectable ones, they are highly paid ones, and they are respected just like doctors. So now why to choose embryology as your profession? So India, we have already more than 5,000 IVF centers. They are already operating with a team of 3,000 embryologists, and they are also not trained enough to handle all these technologies. Each center needs at least 2 to 3 embryologists. So you can see there is a huge gap between the embryologists and the IVF centers. Also, currently we are doing around 250,000 cycles a year, and we have a scope of around 600,000 cycles. So it is almost the double the number. So about, I won't go into Indra. First I want to just show. So this is earlier, it was regulated by PCPNDT. So this PCPNDT is the same organization which takes care of the ultrasound centers or the obstetric clinics. So now this has been taken care by ART. Proper body has been formed with the national registry and state boards. So now they are operating all this, and they have given the guidelines that who can be an embryologist. And if you can see the yellow marked highlighted area, so embryologist is the one who is postgraduate in clinical embryology. Actually, it was first said that only clinical embryology, students who have done clinical embryology, MSc in clinical embryology, can practice embryology. But now that has been revised, and now it is said that any postgraduate in life sciences can practice embryology, provided he has done 1 year of course, compulsory on-site course in from a reputed or a authorized IVF center. So now Indira IVF has, Fertility Academy. It is not new actually. So we have been working since 2 years. And earlier we were working only on the training part of the doctors and the embryologists who will be the part of Indira IVF family. But now recently we have opened our doors to the whole society. You can anyway, if you are interested into these courses, you can always opt for these courses. And all the information, we have one counter outside, so you can have the brochure from there. You can have we have the QR code, you can scan and you can have all the information. I would suggest you to Google what infertility treatments are. At least write, Google a word clinical embryologist and scope of clinical embryology. You will get a lot of literature. There's a lot of scope not only in India but abroad as well. So clinical embryologists, they can work as a researcher, they can become quality inspectors and auditors, they can work as a clinical embryologist in some setup, or they can, very interesting to note that they can have their own IVF center as well. So, and they can have a chance to work abroad. There are a lot of prospects in abroad as well. So with this, I would give a rest to my session. And again, appeal to you, please do Google down what clinical embryology is. If you want any inquiry, our counter is outside. You can please take the brochure from there. You can contact us, you can call us, you can email us. We are always approachable. Thank you.
So thank you very much. And I suppose with regards to the academics, obviously the biotechnology is a huge field, and might be this is one clinical application which generally people don't think that how diverse the field is. Today morning when the guests, all our dignitaries were giving small talks, inaugural speech, the things which were mentioned about the future technology, artificial intelligence, and so many things, we could see sitting on the chairs that this all is actually being implemented in the live parts of the clinic. And we are also into artificial intelligence along with infertility clinic and integrating different technologies which are biotechnologically oriented and is actually live in application at India also. So there's a huge scope. Possibly this start of a lecture is out of syllabus because not many must have thought about this kind of session or a future of clinical embryology. But it's just a spark for you all to think on what else can also be there as a hidden careers. And I suppose you have a very good team of dignitaries and professors to guide you to that. So with that, I suppose we'll end up our session, a quick and short introductory to the clinical embryology. Thank you very much.
Hello.
Thank you, Dr. Ritesh and Mr. Naval. So if someone have any question, they can ask the questions. Yeah, please, ma'am.
I would like to add to it. One of my students last year, she was so motivated to do MSc in embryology. She went to main campus, Manipal Academy of Higher Education, and right now she is pursuing a course in embryology. So I am sure in days to come, you'll get so many students will come, and if possible, you are invited to Manipal University, Jaipur for a lecture. And I'm 100% sure this program of yours will get large number of intellects to join the team.
Yeah, thank you, ma'am. And just to add just one thing before I leave, I really welcome anybody, anyone of you who's really keen and interested. I invite, we have around 112 branches pan-India. You just give us a call and we will arrange your lab tour. So that would be arranged. You can actually see what is the role of an embryologist, how they work, what is their job responsibility. So you are most welcome at any of the Indira IVF centers. Right. Thank you.
Thank you very much once again, Dr. Ritesh Agarwal and Mr. Naval Shah for this wonderful lecture. And I'm very hopeful that the students, those who have come from different part of the India, they would definitely be benefited from your talk and they would pursue their career in clinical embryology in future if they will be interested. So now our next speaker is Professor Karl Kuchler from Medical University of Vienna, Austria. And I would like to request Professor Karl Kuchler, stage is yours, sir. Thank you very much, sir. Ms. Kinjal, please come on the stage to give a brief introduction about Professor Karl Kushler.
Thank you. Thank you, Tikam. Can you hear me in the back?
Hello everyone, I would like to introduce our first keynote speaker, Professor Karl Kushler, Professor of Molecular Genetics at Medical University Vienna, Max F. Perutz Laboratory, Department of Medical Biochemistry. He did his university studies, Technical University of Graz. And he has more than 38 years of research and professional experience under his name. And he has received multiple honors and awards like Postdoctoral Fellow of the Alberta Heritage Foundation for Medical Research 1996 and Novartis Prize for Biology, Lifetime Honorary Member of the Austrian Society of Biochemistry and Molecular Biology. 2011 winner of the Science Slam contest at the annual Open Day of the Medical University of Vienna, and many more. He has main research interest in genomics, host pathogen interactions, drug resistance, human fungal pathogen such as Candida species. And he has more than 130 publications, including papers in peer-reviewed journal reviews, book chapters, editor for several textbook and special journal issues. The cumulative impact factor of all publication is currently about 600. With this, sir has a vast experience in management, congress, organization, and professional activities and international activities. He has peer reviewer for scientist journals, including Cell, Nature, Molecular Cell, PLOS, Genetics, PLOS Pathogens, and many more, which has a high impact factor. He has been a member of the advisory board, Cellular and Molecular Life Sciences, member of the editorial board, Molecular Biology, Microbiology, and he is currently a member of associate editor and editorial board member, Frontiers in Microbiology, System Biomedicines, Pathogens and Diseases. He has experience in international science funding and policy making and advisory boards. With this, I would like to invite respected keynote lecturer to address this conference with his remarkable words. Professor Karl Kushler. Thank you.
Thank you very much for this kind introduction. Okay, so thanks again, Tikam, for inviting me. It's a real pleasure. And, you know, again, having seen all the students who are so eager to do science, this is a wonderful future for India, and I think you should really build on that capacity that potentially you have. So I am at the Medical University of Vienna, and my interest is really studying infectious disease, and in particular, fungal pathogens. And the COVID pandemic has really mercilessly shown how unable we all are, the healthcare system, to deal with rapidly emerging pandemics, and even with recognizing the potential of other pathogens to cause pandemics. And this is what I would like to talk about today a little bit, by showing you that there is a fungal pathogen that is really critical in India. It has spread all over India. It's causing massive problems in hospitals and in ICU units, and I will tell you why. And we have identified a new regulator that we think is a reasonable drug target to develop antifungal drugs. Now, the first part, it will be rather general, and for many of you may be boring, but I really want to alert you, and I want to raise your awareness that multi-drug resistance is not an isolated problem. It has become a global problem, and I will tell you why. So, for those of you think that cancer is causing the most lives in the world, this is not true, actually. Actually, infectious disease causes the most cause of death. 30% of all causes of death are caused by infections, all different kinds of infections. And about the same number is caused by circulatory disease. So just not eating or having a lifestyle that's not properly healthy. And in 2016, and the numbers haven't only gone up a little bit, but relatively they have not changed, there were about 57 million deaths, and 20 million of which were coming from infections. And this was caused by viruses, bacteria, fungi, parasites. And many of these pathogens have the propensity and the ability and the potency to cause multi-drug resistance or antimicrobial resistance. And that actually is also another amplifying factor for pathogens to go into pandemic infections. So I need to put this up because I don't know if you have seen these figures on the WHO website. It's really disheartening to see that the major cause of death in the age group from 0 to 5 and from 5 to 14 is basically infections. Of course, in the less privileged parts of the world, there's much more because Medicare and healthcare is not that high end. But nonetheless, overall, over the entire world, infections are really the major cause of death. And this point here should also give you, and I thought about that yesterday when we were driving through Udaipur, road injuries. Most of the kids are actually getting run over by a car or by a truck or by a bus. And this is in the age group 5 to 14, the #1 cause of death. And clinically, also the reality is actually much more gruesome because in the clinic or in intensive care units, you hardly have any single infections by pathogen. Most of the time you have double infection or super infections. And this is a case example, and I'm showing this slide here because of SARS-CoV-2, that co-infections of SARS-CoV-2 and fungal infections causing letalities of 90%, 85 to 90% of patients will die if they contract co-infections with SARS or, for instance, with fungal pathogens or with Mucor or with Candida auris. So that really poses the urgent need to have a really as rapid as possible, and species-specific diagnosis of the pathogen causing these super infections to just have a chance to use the proper drug and the right drug. And as I have said in the beginning, antimicrobial or multi-drug resistant has become an epic problem. And this is no overstatement, this is not exaggerating. For some pathogens, bacterial as well as fungal pathogens, basically people are running out of antibiotics. For instance, gram-negative bacteria like Acinetobacter, for 40 years there has not been a new antibiotic to treat gram-negative infections. Now this has improved a little bit, but the matter of fact is that a record number of countries, and this is from 2020. Oh, Ranuka, you want to talk to me? No. Contribute data that are disturbing rates of clinical resistance in infectious and ICUs. I mean, you may not believe it, but right now in Austria, which we consider a pretty highly developed country, we cannot get antibiotics for childcare infections. I walked through the old town yesterday. There was this little shop. They had all antibiotics that they could possibly use. But in Vienna, for instance, it's really hard to get the right antibiotics. Also caused by the war with Russia and Ukraine. But seriously, this is totally unacceptable, right? Because you need to make sure that infections can be treated. Otherwise, you're going to run into true disaster. And as I said before, so this can actually drive both the duration and the amplitudes of pandemics. So unfortunately, this is a vicious cycle, and all of you and me and us contribute to that. So basically, we need, and there have been recommendations for proper antibiotic stewardship, meaning there are rules that govern the use and the prescriptions of antibiotics. In some countries, for instance, you can get antibiotics without any prescription. That's terrible. That's totally unacceptable because this is, again, a point at the parameter for driving resistance. And that's why the WHO has declared AMR and MDR as the top 10 health threat for humanity for the next 10 years. So this is really a #1 health threat. And the other thing is that has not been appreciated, the costs of healthcare for treating infections, and particularly trying to treat resistant infections, reaches unsurmountable amounts. This is so expensive that many economies cannot even afford this. So in fact, AMR and MDR threatens the success of modern medicines in almost all areas. Now, just give you an idea, for those of you who do not know, the global human antibiotic consumption, including fungicides and antifungals, in 2018 was about 40 billion daily defined dose. So this is a dose that's specific for each antibiotics, and based on that, you can calculate how many antibiotics were used. And on that, so there's about 2,000 tons of chemically pure substances used in our antibiotics all over the world, 2,000 tons. This seems a lot, but in fact, it's not a lot. When you compare, and when you realize that many of the antibiotics that are used in medicine are also used in veterinary medicine. So most of the like aminoglycosides, beta-lactams, fluoroquinolones, tetracyclines, they're really important in the clinic for human treatment, but they're also used in veterinary medicine. And that's a real problem because the antibiotics are less pure than for human consumption, and most of them go into the environment. And just to give you a picture here, the antimicrobial use in agriculture and in crop and animal food production per year, and this is a purely environmental trigger of antimicrobial resistance, in mass production of food animals, there's about 125,000 tons of antibiotics used for animal production, and about 2 million tons for herbicides, pesticides, fungicides, and all of that is in the environment. And our environment is basically a niche for almost any living organism, bacteria. And due to this heavily contaminated soils, you drive a persistent and continuous selection of resistant microbes that can at some point infect humans. Now, as I said in the beginning, there is viral infections, bacterial infections. We're not going to deal with this. My focus will be on fungal pathogens. And fungal pathogens can grow and affect any body niche. It doesn't matter where you look at. It can be your head. It can be an epithelia, barriers, fungi can be everywhere. And we would probably not live without fungi because there's about 1.5 million fungal species. Some people say 3 million fungal species out there in our environment, and only 150 are pathogenic to humans. Many of the fungi that are used in biotechnology have a long-standing track in producing really medicine and antibiotics and other stuff. But these 150 pathogenic fungi, and only 50 of them claim about 1.5 million lives per year. And still, getting funding for fungal infections is basically close to impossible. Nobody really has a lot of interest in funding in fungal infections, at least in Europe and in some of the developed countries. Everybody cures cancer, but nobody really is interested in infectious disease. And so these fungal pathogens are actually posing lifelong threats for us because on the one hand, they live with us. Our immunity has learned and co-evolved with these fungi, particularly with Candida. And even with Candida species, if you are immunocompromised and you are in the ICU, 40% of the patients will die. Cryptococcus is right now the #1 killer in HIV patients. 750,000 people, HIV patients, just die from cryptococcal meningitis and infections. So it's hard. So these are facts. It's hard to deny the relevance and the importance of fungal pathogens. Also, when you compare the morbidity and the burden between fungal pathogens and the most prominent bacterial pathogens like Mycobacterium, you see Mycobacterium is still among the #1 killers as a single pathogen, 1.2 million per year, but the death rate is actually, let's say, very high, but it's nothing compared to Candida. Most all of the Candida fungal pathogen infections have death rates that range from 25–30% to 90%. So this is a staggering number, and so that means we're just not able to really recognize it early enough and treat it in the most efficient way. Now, these are some pictures how this looks. So this is Candida species. This is a little kid having sore. It's not life-threatening, but the quality of living is not very high, as you can appreciate. This Aspergillus, every one of you inhales about 10,000 spores per day into your lungs. Mortality of invasive aspergillosis used to be 90%. Now it's come down to 30–40% with some better drugs. But it really goes into the lung, into the brain. It basically eats up organs like within a few days. So this used to be high mortality. One of the fungi with 100% mortality, so far I don't know of anyone who survived it, is Cerosporium. So there's a black fungus that goes through the lung, from the lung to the brain, and then in the brain it starts to grow, and you only end up seeing this in the pathology when all of the ventricles are basically filled with fungal biomass. And then in South America, for instance, another one is soil-borne pathogen, Paracoccidioides species. Farm workers and workers on the farm are really affected by that because they inhale the spores, and this is what happens. You get like ball-sized growth fistulas and gangrenas, and these patients have to be on really toxic antifungal drugs for 6 months to a year just to have a chance to survive. And in addition to all these pathogens, there's a new on the market now, and it really shows how ignorant we are. Because it started in 2009 in Japan. Here, there was one little tiny ear infection of a woman, and that was caused by Candida auris. And Candida auris is almost like Candida glabrata. It's a haploid pathogen, looks lovely and neat and yeast form, and you won't think that this can cause so much trouble. Now, within 10 years, Candida auris has spread from Japan all over the world. North America, South America, Africa, the southern part, and of course, as you have said, India is a real issue with Candida auris, but also Europe. And now the numbers in Europe are heavily increasing. And they have been isolated, sort of evolved in isolation. So there's 5 clades, clade 1, 2, 3, and 4, and there's a new clade from Iran that has been sort of making their way on this epidemiology web. So what is Candida auris? And I need to tell you this. This may actually be one of the first fungal pathogens, or if not the first, which is thought to have moved the host range due to global warming and the climate change. So it was thought that Candida auris is actually living in wetlands. So what's the hypothesis? And then with drying up of these wetlands, it jumped over to birds. And this fits very well because this Candida auris can live at 42 degrees Celsius. They love high salt and sweat and stinky fatty acids to grow on. And so they jumped over to birds, and then they went to agricultural rural areas to finally then being transmitted in hospital settings from patient to patient and from healthcare workers to patients. And this idea was actually then really verified. Anu Chowdhury at University Clinic in Delhi, she actually did get permission to go sample to an area of Andaman Islands, which is completely remote. There has not been a human ever since this is on this planet. And they sampled in Andaman, South Andaman Islands, and they actually found Candida auris being there in the salty pools in the ocean. And a couple of years later, she actually also showed that the agricultural environment, like fruits, can be a sort of niche for Candida auris, particularly apples. So the reason for that is when apples are harvested, they're not completely ripe. So they are being sprayed with the wax. And this wax gives them the shiny, beautiful, rosy red or whatever color. But this wax contains herbicides, pesticides, and fungicides, just to keep the apple for a longer time fresh and such that they can sell it. And this is a reservoir for Candida auris sitting on these apples to develop resistance because you have a permanent exposure to antifungal drugs. And this is how you evolve resistance and select for resistant ones. And so next time you buy a not organically grown apple, maybe you want to think twice if you bite into it, because it may contain some auris. And then with that, with the human transmission, and then of course easy in the hospital. So this is the reason why the CDC and the WHO rank Candida auris now as the top 3 priority fungal pathogen. And also the CDC declares this as an urgent health threat for the next 10 years. And there's 3 major problems. The first one is they are super glue on the skin. They stick to your skin like super glue. They don't like to be in the body. They stick to the outside of your skin. And patients colonized by Candida auris, there's so much fungus there. If you take a spatula and scrape it off, you see a white coat on your spatula. So it's heavily, heavily growing, as you will see in a minute. And so this skin adhesion makes it really challenging to avoid skin-to-skin transmission in the clinic, because when a patient is colonized, or even a healthcare worker is colonized, touching a patient, and then just skin-to-skin contact would be enough to transmit. And the real problem is, and this is what's just from the slide before, 90% of all clinical isolates are completely azole resistant. You cannot treat this fungus with azoles, particularly not fluconazole or ketoconazole or bosaconazole, whatever is out there, so they're really resistant. 30% And amphotericin B is an antifungal drug that's extremely toxic. So sometimes you have the choice, patient dead or fungus dead, because amphotericin B is really severely nephrotoxic and hemolytic. And so that's why ampho B is usually not given, but this is the last resort antifungal that's being used. And 2 to 8% echinocandins are being resisted, and that means that even these echinocandins that are first-line preemptive therapy cannot be used. Also, in some countries like here, echinocandin is extremely expensive. We're talking here in Europe, 800 to 1,000 euros per day of treatment. So this treatment is more costly than some anti-cancer treatments. So of course, we became interested in studying this, and so we have developed a native human skin model. So this is basically belly skin we get from the surgery department. And when you look here at this native skin, which is undamaged, you see that the fungus always grows. So the human skin has typically these indents and and they really grow like wheat in there. They do not enter, they are unable to enter, but they grow crazy. And this is because they have enough nutrients and other goodies to live there. Only when you inflict wounding or wounds into the outer skin, for instance, when you scratch yourself heavily or you have needles, and we do this in the lab by needling, you can see that Candida auris is able to penetrate through stratum corneum to the epidermis and dermis. And then inside this human skin, they also start to change their morphology. And this morphology change is really fascinating. And this is sort of, let's say, it's not only Dr. Jekyll and Mr. Hyde, it's an incredibly pleiotropic continuum of a single unicellular eukaryote. So this is a clinical isolate from a patient, and then all of a sudden it turns into a so-called white phase cell, but it can also be in equilibrium with brown cells, and these brown cells are still drug sensitive. But only these drug sensitive brown cells can spin out the drug resistant cells. So we are talking here like something like 50 shades of brown rather than gray. And you can see this is probably a continuum, and many of the steps are reversible depending on the host environment and the host immune defense. They can also filament, and this has been argued for quite some time now, whether or not the filament, and they can form filaments and hyphae very nicely. Now you see this in the lab on an agar plate when you just take a brown colony, you stain it with floxin, which is a dye that stays brown cells that become red, and you see the white pimples coming out here. And reversibly, by contrast, if you take a white cell and colonize it on a certain medium, you can see that the brown and the pink-brown pimples come out of this white colony. And the frequency of this switching is very high. We're talking 10 to the minus 2, about sometimes 10 to the minus 1. And this is also happening most likely in the patient. So what I just said is that the brown, this light brown cell type is really associated with drug resistance. And drug resistance is, as I said, first of all, a problem. Second, it's never ever caused by a single mechanism. It's extremely complex, it's dynamic, and it's caused by different biological phenomena. So transporters, my old love, they're causing drug resistance in many different systems, in many different bacteria, in cancer cells, and fungi. But also genetic rearrangements, copy number variation. Many pathogens grow in biofilm. When you have a catheter inside your veins and there is a thick biofilm forming, so this biofilm is usually resistant to antifungal drugs. But they can also rewire their metabolism and then cause drug resistance. The classically, of course, mechanism is mutation in drug targets, meaning you need to have higher concentrations to get mode of action. And finally, the cell wall and lipid stress is also a cause of drug resistance. And a former student in the lab, she has actually discovered that white-brown switching is also intimately involved in MDR development. But the overall mechanisms are not known, despite the fact that they are multifactorial, complex, and dynamic. So we don't understand them. That's why we started to study them. So what you do as a molecular biologist, you do a very simple experiment. You do transcriptomics. You do RNA-seq. In other words, you take brown cells and you take brown resistant cells, and then you isolate the RNA after treatment and then look which genes are regulated. And we took 3 different isolates. One isolate which has barely any clinical resistance, so they behave like almost sensitive strains, and 2 other strains which have from medium to very high level of resistance. And as you can see, depending on the resistance level, the weakly resistant ones cluster basically like the sensitive cells, so they're pretty much the same. But when you get to higher resistance, they become completely different pathogens. And if you then look at the Venn diagrams of the genes that are regulated or not, you can see there's a cluster of about 500 genes. There's 500 genes that is linked to amphob resistance. And there's 4 major mechanisms that are involved from the genes. So first of all, there's adhesion and biofilm formation. All of the genes you see here are upregulated when you go to the resistant strains. Chromatin. Chromatin is extremely adaptable and can actually change adaptively and transiently resistance mechanisms. And for the chromatin lovers, many of the chromatin modifying genes and acetyltransferases, deacetylases are found in this GO term. Transport, I have already mentioned. All classes of transport that we know in fungal cells are linked to this resistance. And finally, lipids. Lipids are really at the core of drug resistance because all of the ergosterol synthesis genes are involved in this resistance. So now if you were a student and starting with this project and find out which gene is actually really causing it, you would have to knock out all of these genes, make combination mutants, make double mutants, triple mutants, and then figure out what the phenotype is. So we decided to do something else. We decided to take a proteomics approach. And what we did, we took the same cells that are sensitive and resistant, so light brown and dark brown, and cultured them overnight. We, in addition, added amphotericin B, so you did the treatment as well. And then we make total protein extracts, put this into LC-MS, and then we took all of the proteomics data and overlaid them with RNA-seq data. So, and this is the outcome. This is after quite a bit of work, and it's so basically sort of a summary slide from all of the transcriptomics data we had. We overlaid the genes that were regulated with the proteomics data we had, and then we ended up with only 14 genes. So 14 genes is actually pretty cool because that means you only need to do 14 knockouts. And we named these genes regulated by amphotericin B, RBAs. RPA 1, 2, 3, 4, 5, 6, 14. And we have some more that go to 25.
Hello? Only few minutes. Sir, I'm here. 2 minutes.
2 minutes? Okay, good. Then we have to speed up. And one gene turned out to be very interesting. This was RBA1. And then we were also looking for a transcription factor that would be regulating RBA1, and this is ROM1, and we called it regulator of metabolism. Now, ROM1 is a classical hallmark BCP transcription factor that's conserved in all fungal species, not only in Candida species, but also in Aspergillus, Mucor, whatever you look at, it's conserved as the same domain organization. Now, ROM1 is a direct regulator of RBA1, and RBA1 is an enzyme that converts carbon dioxide to hydrocarbonic acid, and that feeds directly into the TCA cycle, into energy metabolism. And at the same time, hydrocarbonic acid activates morphogenesis by driving a cyclin AMP and EFG1 morphogenesis. So, of course, we did the knockout and asked what is happening when you do the knockout. And so again, when you do the knockout to RNA-seq with the transporter of resistance versus ROM, you see all of our favorite transporters are regulated, and all of our RBAs are also regulated when you just overlay again the proteomics data with the sequencing data. And so we have here about only 10 genes left to validate. And now when you look at the phenotypes of the cells, when you take away ROM1, you can see that in these cells, basically, you have a very uniform filamentation. They cannot form longer hyphae than 4 to 5 segments, whereas in the wild type, you have these clumpy, different size yeast form cells with filaments. And you can see this in the facts. This is a very uniform population. Now, the most interesting part, and yeah, I need this minute, is when you then look at, take a white-brown competent strain and you remove ROM1, you can see that in ambient air, has a severe defect in white-brown switching. But if you do this under carbon dioxide, there's a complete inability to switch. So of course we tested this, and I didn't believe this data. I had the students do this 10 times. before I believed it. So if you just take now wild type and ROM1 mutants and do an M4P MIC testing or IC50 testing, you see that in ambient air, there's a fourfold difference in the MIC of ROM1 deletion mutant versus the wild type. But when you put this in carbon dioxide, it's completely abrogated, so the resistance phenotype is gone. So this tells you ROM1 somehow is involved in carbon dioxide sensing. So we also tested, of course, cell wall phenotypes, and ROM1 also controls cell wall changes. This is the lipid membrane, glucan, and the mannan. This is not changed, but the mannan is actually really heavily upregulated, implying there's also some interaction or changed interaction with the immune cells. And this is indeed the case. So in vitro, you see a different adhesion. So the cell wall has changed, and that means they adhere much better to plastics. So the plastic adhesion is dramatically up. And when you do this experiment then in vivo, you take primary macrophages, you treat them with Candida auris, you can see this is even more sticky than the resistant parent strain. And conversely, you would expect that they are better phagocytosed and then they are better killed. So the killing is much higher. So to prevent from getting me off stage, I will just sort of put you this sort of into physiological context. Can this be reality? Well, if you think about your skin, on the outside of your skin, you have 0.01% carbon dioxide. So under these conditions, ROM1 is active and is driving RBF1 that produces hydrocarbonic acid and at the same time regulates carboxylate metabolism and cell wall remodeling. But when these cells go into deeper layers, like into the dermis, even along hair follicles, you actually have a different carbon dioxide. You can't see this because... So in the dermis, there's like 5% carbon dioxide. And so that's enough carbon dioxide to just dissolve it in the tissue. That means you don't need RBF1. So RBF1 is being suppressed. And this suppression via other RBF1s actually causes, again, changes in filamentation and changes in fitness and survival. Yeah. So I'm at the end. So I just acknowledge the co-workers. So Sabrina, really started most of this work, and I have a fantastically hardworking student from Vietnam, Phan Can Trinh. So all of the data I presented you today were done by him. Philip Benninger helped with the in vivo models. I didn't show you the data, and also Runik, a postdoc from India, and Semih, a student from Turkey, helped with some of the phenotyping. We have world-class proteomics in Vienna, Marcus Heidel, who did all of the proteomics. We have this dermatology department, with whom we collaborate. And of course, we have a very long-standing and active collaboration with Neeraj Chauhan at Hackensack in New York, and of course, with Anu in Delhi, who is providing us with all of the clinical isolates of Candida auris. So with that, I'll be happy to take questions.
Thank you very much, sir. It is a great presentation. And use and misuse and overuse of randomly using the antibiotics leading to this kind of a biggest problem in the world. Biggest problem in the world. Misusing of the antibiotics. That's what your first presentation. The second one is one of the biggest one, Candida auris, is one of the bigger problem. Oh, I'm sorry, I can't hear.
Sorry, what was the first question?
No, it is a great presentation. One of the best presentation I ever saw in this, especially in the MDR phenotypes and how they come and how we fight. Animal and plants warfare. Keep on, and we are running out of the time, so we will keep on moving. And if you have any question, one question, only one. Yes, please.
Hello.
Yeah.
I have one simple question, like regarding to the development of infection like Candida or viral. We can say like types of cancer could be developed. So we could consider that development is a kind of drug resistance. Like as the drugs is not working, so it can make the mutation inside the genes. Then it can develop the cancer, the different types of cancer.
I'm so sorry, I didn't understand the question, Funedi. Can we talk? We talk after. Yeah, we can discuss in detail. Okay. Go ahead. Go ahead. If
yeah. Yeah.
Okay. Yeah. So this is a very good question, actually. So she was asking whether there's a role in cancer development of some of these microbes that are resistant. Yes, there is actually, but not for Candida. There is at least a clear correlation between IBD, inflammatory bowel disease, and also Crohn's. If you have a sort of dysregulation of your microbiome, you get chronic inflammation. That promotes IBD. For bacteria, Helicobacter is very well known, gastric cancer. So if you have overpopulation of Helicobacter, you are very much prone to get gastric cancer. Your turn, sir.
Thank you very much, sir. And our next speaker, Professor and also called Professor Suresh, and he speaks fluent Hindi as well as English, and he sounds like more Indian rather than coming from Netherlands or Holland or Austria. Madam, can you introduce?
Should I give you a talk?
A big round of applause for Professor Kuchler. It has been a really educational lecture. A big round of applause for him. Thank you so much, sir. I would like to introduce our next keynote speaker, Professor George Van Driem. Professor George Van Driem has been a professor emeritus at the University of Bern. He has written grammars of previously undescribed languages of the Himalayas, that is Limbu, Dumi, Bamthang, Dzongkha. In Bhutan, he conducted a linguistic survey of the kingdom, collecting the first linguistic data on 3 languages previously unknown to science, that is Lokpu, Gongduk, and Black Mountain Monpa. He published 2-volume handbook languages for the Himalayas, a comprehensive history of tea entitled The Tale of Tea, which has now also come out in Mandarin edition, and the new handbook, Ethno-Linguistic Prehistory. A big round of applause for Professor George Van Driem.
मैं बंद करूंगा हो सकता है लेकिन कैसा करना है डुप्लीकेट है यहाँ अब तो ठीक है लेकिन यहाँ से मैं बिल्कुल ठीक है चलेगा। नमस्ते दोस्तों, मैं थोड़ी देर हुई लेकिन अभी शुरू करना है। I'm going to talk about Indo-European, Indo-Iranian, and Burushaski, linguistic intrusions, and the Aryan controversy. The Aryan controversy is something that gets some people really worked up in a froth. It's something that is extremely emotional for some people. But actually, I have a secret to share with you. This controversy only exists within India. people at universities outside of India, and actually people, I think, at most universities within India, do not see this as a controversial matter, but it has been converted into a controversy in public discourse. And sometimes it has been converted into a controversy in public discourse by paparazzi. So actually, prehistory, patrakaron ka tamasha nahi hona chahiye. It is something that prehistorians, paleontologists, linguists, and geneticists have to study in a cool, calm, and collected way. So let me see if I can move on. The controversy made its way all the way to Bollywood, and we have here the story of the invaders of India in a Bollywood film. If you look at these books at the bottom, that I'm showing at the bottom, you see the wheels, which of us are Aryans. This is a compendium done by various scholars, and it is a very good book to be recommended. This book by Tony Joseph, who is a friend of mine, and he wrote this book after 6 years of studying the literature very carefully, understanding it, and then digesting it and communicating it to the interested lay audience. So they did a great service. But these people on the left of this yellow vertical line, they accept the cumulative evidence for what has been called the Aryan invasion or the Aryan linguistic intrusion, which sounds less violent than an invasion. But many people you see are writing books in India to the right of this line, and perhaps to the right of the spectrum in general. And these people argue in a polemic fashion and very passionately against the Aryan invasion. They don't want to accept the Aryan invasion of the Indian subcontinent. And actually, I have good news for everybody. If you come to think of it, why do we have to worry where our ancestors come from, since molecular biology shows us that ultimately all our ancestors emerged from Africa? But there are people who want the Aryans to have come from India. But I have good news for both groups in this controversy. The people on the left analyze the science correctly. The people on the right are not really analyzing the scientific information correctly, and they have misinterpreted some of the scientific evidence. However, they are right nonetheless at another level, because the Aryans too, even if they came outside of India, they came from somewhere, and the ancestors of the Aryans, they came from India. So therefore, the point of the story is that time took an awfully long time. The past, prehistory, took an awfully long time, and many things happened. And the spoiler alert would be back migration. So before modern population genetics, this man wrote a book that I can still recommend to you, even though it is about 30 or 40 years, 35 years old. The book was written by James Patrick Mallory, professor at Queen's College in Belfast, and it's about the Indo-Europeans and where they come from. And although he is an archaeologist, he does not only all the archaeology, but he has read and understood all of the philology and all of the linguistics, the historical linguistics, which he reads, whether it is written in Georgian, Russian, English, French, German. He reads all the literature, and this is really a good book, but it does not include genetics. This is a book that I wrote, and I actually lecture about this topic. But the lecture about this topic takes one semester, and then I assign students many hundreds of pages to read in the specialist literature, including the 1,475 pages in these two books. So they read this in order to understand the ethno-linguistic prehistory of the Indian subcontinent. And I'm going to try to tell you a whole bunch of it in just 25 minutes. So I'll be fast, but I'm going to skip quite a lot of things. But I can give you a lot to read, beginning with that book by James Mallory. This is a language family. It is the Indo-European language family to which Hindi belongs, German, French, Russian, English. And like in biology, a family tree or a tree is determined by the branching by nested innovations, shared innovations, mutations, and you can demonstrate, just as with a Y chromosomal phylogeny, why single nucleotide polymorphisms, molecular mutations that are then shared in subsequent generations. That's what determines, and here with phonological sound laws and morphological innovations, we get the same thing for the Indo-European language family. So Proto-Indo-European splits into Anatolian and other Indo-European, which splits into Tocharian and Classic Indo-European, which splits into Italic Celtic, and Central Indo-European, which splits into Germanic, and Greco-Aryan or Nuclear Indo-European, which again splits into Greco-Phrygian and Satem languages. Satem splits into Thraco-Armenian and North Satem, and North Satem splits into Daco-Albanian and East Satem, which finally splits into Balto-Slavic and Indo-Iranian, Vedic, Sanskrit. So at the top of the tree, we see the languages here in northern India. And this is a well-supported Indo-European, up-to-date Indo-European language family tree, state-of-the-art. What we know about the spread of Indo-European based on old texts, comparative historical linguistic evidence, and the archaeology leads us to believe that the distribution was like this around 2300 BC, with, you see, the precursors of Indo-Aryan in Soviet, what used to be Soviet Central Asia, and then there was a north-south split with Indo-Aryan towards the south and Iranian towards the north. Then these move into the Iranian plateau, and the first people to enter are Indo-Aryans, who move across the Punjab into the Indus, across the Indus, and across the Persian plateau, all the way to the Jazeera in the north of Mesopotamia, where we know that the Mitanni were an Indo-Aryan elite who conquered a Hurrian populace and became the aristocracy, the ruling class in the Mitanni kingdom. We know this from pharaonic Egyptian records and Egyptian libraries which have been unearthed, which describe this movement of the Mitanni. Later, as a result of this, Indo-Aryan and Indo-Iranian enter into the Indian subcontinent, spread across the Iranian plateau, and by 1500 BC we have this distribution, and then by 500 AD, Indo-Aryan has spread all the way across North India, and Iranian has spread all across the Indo-Iranian plateau, and these are then the different Indo-Aryan languages of the north of the Indian subcontinent. We don't see Sri Lanka because we have an Indo-Aryan language there too, Sinhalese. But that's another story, how it got there. Then we have another language family, very prominent in India and very prominent in the world, because with over 300 million speakers, this is one of the major language families of the world. There are 19 languages, some people say 20 because of an argument about two languages or two dialects, how different they are. But the distribution is always, we always think of Kannada, Telugu, Tamil, and Malayalam, these big four literary languages, and we identify the language family with the south of India. But we have northern Indian languages like Kurukh and Malto, and even in the north, in Afghanistan and Pakistan, particularly Balochistan, we have Brahui, which is a Dravidian language. And when we look at the phylogeny of this language family, and we will look at it in a second, we will reevaluate this distribution. Here is Elam. The kingdom of Elam has the second oldest writing system of mankind. The oldest writing system is that of the Sumerians, which dates from 3200 BC. And Elamite writing, which you see in the upper right hand on the top of this urn, or on the top of this jug, you see Elamite, Proto-Elamite pictographic script. It is the second oldest writing system in the world, and this dates from 3100 BC. 2,000 years later, so this is the map, shows you the situation 2,000 years later. The Elamites are still there where they have been, but they used to be across the Iranian plateau. But now, from the 11th century, as we know from pharaonic Egyptian records, but particularly from Babylonian records and also from Greek and Armenian records, the Medes, the first Iranian speakers, move to where you see them on this map, coming from the east. And these are the first Iranian groups. Their descendants may be the Kurds, linguistically speaking. And so we see Iranian speakers coming in at the beginning of the first millennium or end of the second millennium BC. And Elam used to be spread all across the Iranian plateau before this. The Elamite trade network was spread, and we see Proto-Elamite administrative tablets. We find them all in Central Asia, and even in the Indus civilization, we have, from the trade, we have Proto-Elamite writings. So Elamite was spread across this region before the Iranians came and settled on the Iranian plateau. No scholar of antiquity thinks that in the beginning of the second millennium BC, we had Iranians already on the Iranian plateau. They come later because first the Elamites meet the Gutaeans. Only after do they meet Iranian peoples. Then once, here's Proto-Elamite script on the upper right-hand corner, and in the upper left-hand corner. Below this, on the left-hand side, is the Indus script, the script of the Indus Valley. It is not only similar in terms of its typology, but it is also similar in that there is a material correspondence of some of the actual glyphs. So they used related writing systems in the Indus Valley, and the Proto-Elamite used a related writing system. This is the Indus Valley civilization, which dates from the end of the 4th millennium BC. And of course, mature Harappan is when the Harappan civilization was at its most affluent. And this is when it was also at its most vulnerable, because being very affluent, it drew migrants from elsewhere. Why did people start digging in the Indus Valley, archaeologists? It's not because they simply thought, let's dig there. It is because philologists, people working with Vedic texts, the Rig Veda in particular, such as Christian Larsen and Hermann Gottlieb Brunhofer, they looked at all of these old texts. They said, based on these texts, the old Aryans, when they entered the continent and began waging the wars that are described in the Rig Veda against Aboriginal peoples who were there before, or native peoples, we think that it must be in this area in the north because of references such as Haryupia and Vrichivat. So people looked there. The archaeologists looked in the Indus Valley because the philologists and the linguists told them to look there, and only afterwards was the Indus Valley civilization discovered. So even very famously, Binod Bihari Roy in 1928 proposed that the Rig Vedic place Haryupia, where this very bloody battle takes place, and the Aryans come in and slaughter the denizens of the city, that Haryupia could have been identified with Harappa itself. This is a proposal by the Bengali scholar Binod Bihari Roy from 1928. The Aryans are led by Indra, and Indra is the god, the Puramdar, who is destroying the Puras, the citadels of the indigenous people, as the Aryans enter. This is all recounted in the Vedic texts. So it is not something that controversial. There's 2 centuries, over 2 centuries of scholarship, philological, textual, historical, linguistic, and archaeological, buttressing this scenario. But it troubles some people, as you saw in the beginning. But after this talk, I think those people need not be troubled either, because they will find out that they are right anyway, but at another time depth. So everyone is right, simply at different time depths. Airavata Mahadevan compiled the first exhaustive concordance of all Indoscript symbols, and Asko Parpala in Finland has worked on the decipherment. He ended up with basically a model for a Dravidian script. There's a Dravidian language underlying the Indoscript. There's even a character that arguably represents Murugan. You had a group in the Soviet Union in Leningrad under Knorozov working in a very adversarial relationship to this group, but they also did a reconstruction of the Indoscript, a decipherment, and they came up with basically the same Dravidian affinity and most of the same reconstructions. Older than either the Indoscript or Elamite is this primary Neolithic, which goes back to 7000 BC, and that's almost 10,000 years old, and that's on the in the Hindu Kush and on the Iranian plateau, and this is a primary Neolithic. So this is perhaps it's definitely the archaeological antecedent for the Indus civilization. It may even be anterior to the Elamite civilization. Linguists for a long time have claimed, it started with Caldwell actually, that Elamite and Dravidian are related. The evidence that was presented by Caldwell was impressionistic and also based on the inscription at Behistun, which was in Elamite after it was deciphered. But this is Igor Mikhailovich Diakonov in the Soviet Union in Moscow. I met him once in Leningrad before he died. He adduced all sorts of linguistic evidence uniting Dravidian and Elamite as a language family. This scholar, Mikhail Sergeyevich Andronov, I had to read his book in the original Russian edition as a student because the English translation was not yet available. He pictured the Dravidians based on the phylogeny of the language family as moving into the subcontinent from the northwest, and you can see he used the technique, the Russian technique called glottochronologia, which means glottochronology, to date the dispersal of Dravidian based on the structure of language family tree as the Dravidians moved. He also spoke fluent Malayalam. I am jealous, I cannot do that. But so we know when we look at the grammar of Dravidian, we know what is ancient Dravidian grammar and we know what is modern accretions, what is modern innovations. The work by McAlpin and by Stamford Stever have shown us this very clearly. This is the language family tree of Dravidian. You can see that the phylogeny of the tree is rooted in the north with Proto-Brahui in Balochistan towards the top. The connection with Proto-Elamite, controversial for some who would like to see greater evidence adduced than what has been adduced so far. But the rest of the tree is not controversial, and you have Dravidian located in the north and spreading just as Andronov saw it towards the south because of the structure of the language family tree. This is Badri Raju Krishnamurti. There were some people opposed to the Elamo-Dravidian hypothesis, and initially in the 1970s, even Badri Raju Krishnamurti, the famous Dravidian scholar, was opposed to this idea. However, with time, you see, when he first presented the language family tree, it's the same as the previous tree. He only represents it differently, and he switches east and west around intentionally to distract the attention from the idea that the Dravidians originated in the northwest. So, but later, before he died, in his 2003 book, which is the exhaustive and most authoritative book on historical Dravidian grammar, he has mellowed his stance and he is very accepting of the Elamo-Dravidian hypothesis. So his mind was slowly changed and was compelled. He was compelled to change his view in the face of the overwhelming cumulative evidence. Here's where Brahui is located. The Brahui live together in the same place with the Baluch, but the Baluch are Iranian speakers. They speak an Iranian language, but the Brahui and the Baluch have a hierarchical relationship between themselves. Brahui women like to marry Baluch men because the Baluch have higher status. The Brahui seem to be a remnant of the old Indus population, a linguistic remnant. This is what many linguists believe. This man wrote 3 volumes. He lived in Quetta and he spent half of his life living amongst the Baluch and the Brahui, and he wrote these very important studies. Anyone who wants to understand the historical relationship and the social dynamics of this Baluch-Brahui commensal relationship, I highly recommend these sources. They're old, but they are very rich, replete with relevant information. Now, something that Patrick Mallory had no access to because modern population genetics, modern molecular genetics has told us so many things. And what do we find? Well, when the geneticists started looking at the global distribution of language families and language groups and comparing it with molecular polymorphisms, what did they find? This is a group of Swiss and Italian population geneticists. They found this father-tongue correlation. They found the historical...
Professor Suresh,
yes.
2 minutes.
That's good. So they found that this geographical distribution of language families and language groups correlated very well with the genes, and particularly with the uniparental marker on the Y chromosome. And these Y chromosomes spread. We have lots of bottlenecks that date back to the beginning of the Holocene, and Carmine et al. 2015 is a very important paper. We just saw it represented in Benares at the conference, where you have these bottlenecks dating from this time. And many language families correlate very well with these Y chromosomal markers. Here we see part of the story, and the L haplogroup there in the Iranian plateau is what we associate with the Elamo-Dravidian. And I proposed this first in 2012, but because you see it there in the Iranian plateau, but subsequently it moves into India. And this work that was just presented by Sekera and Mustaq in Benares gives all this new molecular genetic evidence showing how L spread into Dravidian groups or with Dravidian groups or with early Dravidian pioneers into the south of the Indian subcontinent. Last is Burushaski. They live in Gilgit, which is where the red dot is, and that is in Pakistan occupied Kashmir. So actually they live in India, but the Burushaski are there. Tibet is occupied by China. This area where the Burushaski live is part of India, but occupied by Pakistan at the moment. And this Burushaski is very interesting because just as Indo-European and Dravidian are separate language families, Burushaski has always been considered to represent a language family all by itself, unrelated to any other human language. Ilya Chashule is the first one to present evidence that Burushaski could be Indo-European. Here it is. He also believes that it is closely related to Phrygian. That is more controversial, but he has presented evidence that, in his publications, of a Burushaski Indo-European relationship at a very deep level. I reevaluate his evidence in this book, which came out last year, Ethno-Linguistic Prehistory. And I talk about the father R theory, where you see only the first hypothesis of Chashule is really supported. That is the deep Indo-European affinity. And you see that the Proto-Indo-European, characteristically associated with the Y chromosomal subclades R1, R1A, and R1B, whereas Burushaski is overwhelmingly R2. So Proto R, where father tongue R would connect them. And if we look at the Indian subcontinent, we see that R comes from the Indian subcontinent. The ancestors of the Aryans came from the Indian subcontinent. The ancestors of many people came from the Indian subcontinent. And all of the ancestors, so many groups came from or came through India, but that is in my book. That is not in this presentation. But R came from India. R2 is found very strongly in the Burushaski, and R1A and R1B, which we find all over Europe and all over the world, including in India, in Indo-Aryan, speakers of Indo-Aryan and Indo-European languages, that spread from the steppe, the Pontic-Caspian steppe to the Central Asian steppe and back into India. It's a back migration. The ancestors of the Aryans, at least the linguistic ancestors of the Aryans, they left and they came back. Last thought is father tongue P. If we look at the bearers of R clades, and then we have Dene Kusunda, and then we have father tongue P, because P on the Y chromosomal phylogeography is ancestral to both R and Q, and all of the Dende Kusunda language or linguistic phylum speakers are overwhelmingly Q, as is everyone in North and South America who is native there. And this too, at a deeper level, came again from the Indian subcontinent. Because the Indian subcontinent, हमारा हिंदुस्तान मातृभूमि है, मानव जाति की मातृभूमि भी है, पितृभूमि भी है. It is really the homeland of many, many peoples on the planet in Eurasia and the Americas. So at different time depths. This is the maximum time depth that we can look at, this, because we can only go further back to a certain point in linguistics and then no longer. Population geneticists can go further back, but linguists cannot go further back because then we have no more empirical basis. Language simply changes too quickly, and that is all I can tell, just a snippet from a two-semester course that I give, and just telling you about the Aryans, and I think there should be no more Indo-Aryan controversy. It is now obsolete.
Thank you, Professor George. It was a very nice, very beautiful talk on Indo-European, Indo-Iranian, Burushaski linguistic intrusion to the Aryan controversy. And you have very nicely brought out all the points, gathered all the evidences to prove this particular hypothesis. Although I'm not from this background, but the audience and me, maybe we can appreciate that this type of work requires deep analytical work. It is a very, very laborious work, and we really appreciate the effort made by Professor George. Thank you very much for your beautiful oration. Thank you, sir. Currently, we are not having any questions. Just one question, please. Kindly be brief as possible, as much as possible.
Shri Chauhanji.
Hello. Thank you. It is time to take the lunch. All are hungry. I congratulate you that you have done a very wonderful job. In each and every pictures, I see that how the languages travel one from another places, including the Europe, Asia, and India, and North India, South India, and Mohenjo-daro and Harappa. I was looking after the pictures, and you have depicted geographically the location of Mita Thal in Haryana, in Bhiwan, Raki Gadhi,
yes,
very close to my village. So the location you showed near about Gurgaon or Delhi, so
yes,
should be okay.
Yes,
It's correct, please. Another point is that you talk about the Indian continent because there are so many students there. You explain the correct definition of about Indian subcontinent. It will be better. Geographically, I know the truth. There are so many students who are not aware that what is the correct definition of Indian subcontinent. And I congratulate and very happy. The issues, very, very relevant issues. Congrats again. Thank you.
Thank you very much. Thank you very much. Yes, Rakhi Gari is very important. We just had a very big discussion in Banaras about terminology, because in some of those publications, people talk about Iranian-related ancestry, but what they mean is prehistoric Persian plateau ancestry, but actually before the Iranians came there, because they're talking about a time depth that is much older. So when people are saying something did not come from the steppe, but maybe came from the Persian plateau, they're maybe pointing to a Lama Dravidian. So this is a very complex discussion and a very wonderful one. I wish you had all been in Banaras, because we discussed it. It was a very beautiful convivial conference, ADNAT, that I just attended at Banaras Hindu University. It was wonderful. But thank you so much for your valuable points. Thank you so much.
A big round of applause for Professor George Van Drim for making this lecture fruitful and prolific.
वॉलेंटियर्स, एक चीज का हमें ध्यान रखना है कि अभी लंच के बाद में सारे पार्टिसिपेंट्स को वापस ऑडिटोरियम में ही लेके आना है। ये इंश्योर करना है कि कोई भी पार्टिसिपेंट बाहर नहीं जाए, ठीक है? हमारे टेक्निकल सेशंस चलते रहेंगे और पोस्टर्स और ओरल प्रेजेंटेशंस भी होगी पैरेलल में भी, ठीक है? दुष्यंत, कविशी, सबको बता दो।